Coming to synaptic inhibition, there are two types of synaptic inhibition: postsynaptic inhibition and presynaptic inhibition. Postsynaptic inhibition, it depends upon the delivery of an inhibitory chemical uh, by the incoming axons which hyperpolarize the subsynaptic membrane. It is subdivided into direct or indirect inhibition. Direct inhibition of the postsynaptic membranes occurs by development of the EPSP. For example, afferent fibers from the muscle spindle in skeletal muscles are known to pass directly to the spinal motor neuron of the motor unit supplying the same nerve in a stretch reflex. Impulses in this afferent supply causes EPSP, which summate to produce propagated response in postsynaptic motor neuron. But at the same time, IPSPs are produced in the motor neurons supplying the antagonist muscles. This latter response is mediated by branches of afferent fibers that end on Golgi bottle neurons or interneurons. So these interneurons in turn secrete inhibitory transmitter, that is glycine, at the synapses on the proximal dendrites of the cell bodies of the motor neurons that supply the antagonist, and this is example for direct inhibition. Therefore, activity in the afferent fibers from the muscle spindles excite motor neurons supplying the muscles from which the impulses come and inhibits those supplying its antagonist, and this reflex is called as reciprocal innervation. There can also be indirect inhibition of the postsynaptic membrane. This can occur because of the effect of previous postsynaptic neuron discharges. For example, the postsynaptic membrane can be in a refractory phase, and it can be refractory to the excitation because it has just fired and it is in the refractory period. It can also occur due to after hyperpolarization, where the membrane potential becomes more negative, and it can also seen in Renshaw cell inhibition or negative feedback mechanism, which is also called as a negative feedback mechanism. Okay. So this diagram showing the postsynaptic inhibition here. The postsynaptic inhibition can be direct when the interneuron secretes inhibitory neurotransmitter, or postsynaptic inhibition can occur because of the effect on the postsynaptic membrane, whether when it is there in the refractory period. Okay, so then the cell is not cannot be excitable. Renshaw cell inhibition. In this condition, motor nerve gives off some collateral branches as they traverse the spinal cord towards the ventral root, and these collaterals make excitatory synaptic connections with interneurons called Renshaw cell, situated in the ventromedial part of the ventral horn. These Renshaw cell in turn send axons which make inhibitory synaptic connections with the motor neurons, and Renshaw cells are stimulated by acetylcholine. So this diagram showing the Renshaw cell inhibition, where you can see the anterior motor neuron. So when it discharges, it sends some collaterals to the Renshaw cells which are inhibitory in nature. They, by negative feedback mechanism, they can inhibit the other motor neurons by producing negative neurotransmitter, and these cells becomes inexcitatory to the other impulses. Okay? And this is example for the postsynaptic inhibition. Presynaptic inhibition. This type of inhibition occurs at the presynaptic terminals before the signal ever reaches the synapse. Presynaptic inhibition occurs because of the failure of the release of excitatory neurotransmitter substance from the presynaptic axon terminal. This occurs in synapses where inhibitory neuron synapses with the afferent fibers of excitatory neuron before the later synapses with the efferent neuron. In other words, presynaptic inhibition occurs because of the axo-axonal synapses. Okay, so this is the diagram showing the presynaptic inhibition. Here, this is usually the excitatory, and this interneuron It forms the axoaxonic synapse with this excitatory neuron, which is usually inhibitory. It secretes inhibitory neurotransmitter, thus it is going to suppress the excitatory impulse on the motor neuron. Activation of presynaptic receptors reduces the opening of calcium channels directly or increase chloride and potassium conductance, so which indirectly causes or reduces the calcium entry, and this decrease of the size of the action potential reaching the excitatory ending. So the amount of excitatory transmitter released is reduced. The inhibitory transmitter at the exoaxonal synapse is usually GABA. Acting via GABA A receptors, it increases chloride conductance, whereas GABA A receptors via G protein produces an increase in potassium conductance. Other transmitters also mediate presynaptic inhibition by G protein mediated effects of calcium on uh, G protein mediated effects on calcium channels and as well as potassium channels. Note that there is also a phenomenon called as presynaptic facilitation, which is produced when action potential is prolonged and action and calcium channels are open for longer period. Therefore, repolarization gets delayed and allowing the repolarization to continue. It is mediated by serotonin. Serotonin released at the exoaxonal endings, it increases intraneuronal cyclic AMP, and this results in phosphorylation of one group of potassium channels. Potassium channels closes the channels and slowing the repolarization and prolonging the action potential. There is also a phenomenon called as feed-forward inhibition. It is seen in the cerebellum. In this type of inhibition, a neuron is connected through two pathways. One is excitatory and another is inhibitory. For example, in cerebellum, stimulation of basket cells produces iPSP in Purkinje cells. However, the basket cells and Purkinje cells are excited by the same parallel excitatory input, and this arrangement is called as feed forward inhibition. This helps to prevent the duration of the excitation produced by any given afferent impulse. Okay, so you can see uh, the the basket cells, I mean, they are mainly excited to the inhibited to the Purkinje cells, but some of the afferents which are going from the basket cells, they themselves stimulates the Purkinje's fibers, and the basket cells usually it modulates the excitatory output from the Purkinje cells, and this kind of